Hi, everybody. Welcome to this next episode of MCS Science. This one is for my forensic science students. This one is about DNA fingerprinting, very useful tool. To kind of introduce DNA, everybody should understand that it is deoxyribonucleic acid. So it is a very important genetic molecule. It is present in all of our cells except for mature red blood cells. They don't tend to have DNA because they don't reproduce on their own. It is true that no two people have identical DNA, even though it says here except identical twins. Um, the fact of the matter is, in the last 10 years or so, we've discovered that even identical twins don't have the exact same DNA because DNA changes can compile through the life of a person, and they can actually become more and more different as they age. It's kind of an interesting concept, but it's a fact. So if you look at this, the images that are down here, you can kind of see how identical twins would form over here on the left. If they're monozygotic, they would be identical, which means one sperm, one egg. They went through cell division, and as they were going through cell division, those two cells, which were supposed to stay together, split into two distinct entities that then both went through cell division and became humans. Fraternal or dizygotic means two eggs and two sperm. So there is no real DNA connection more than any other sibling in the family. So if you look down on the right hand side, you can see what's called the DNA fingerprint, or in this case, it's actually a really long DNA fingerprint. It's got a lot of markers in it, and you can look at it and see that some of these individuals, A1 and A2, don't share the same bands in this gel, so they are not identical twins. B1 and B2 appear to share the same bands all the way down. Those are probably identical. C1 and C2, same all the way down. And then D1 and D2, there are differences between them, so those two are not identical. DNA fingerprinting, also known as DNA profiling, which is a better term for it, it's used to take samples from a crime scene and differentiate between them. The process was invented by an English scientist named Alec Jeffreys in 1985. And initially, it was fingerprinting and only using a few markers. Basically, what you do, you take samples, you put in restriction enzymes, which are a special enzyme that can cut DNA at specific sites, and you mix them with the samples, and then you run them on what's called an agarose gel. And after some period of time, they separate out on the gel according to size, and then you compare them. If you find the same exact DNA at a crime scene as you do in one of your suspects, then you have evidence that that suspect was at the crime scene. So DNA fingerprinting can be used for a lot of different things. It can be used to help solve crimes by identifying victims, but also by linking suspects to crime scenes. It's a very strong form of evidence in terms of, the court of law. Initially, it wasn't really well accepted because people didn't understand how it worked, but nowadays it's become much stronger in terms of its evidential weight. DNA can be used also to determine paternity and parentage. So if in the case of a mother who didn't know which father was, the father of her child, you could check the DNA of the children and compare it to both the mother and the father. And like in this image here, you see that D1 has bands from both mother and father, whereas D2 has bands that only match up to the mother here. Though a couple of these bands appear to match to dad, this one here does not match. If you look, it's just below there. So this individual here, D2, did not come from this father. There are a couple other things here. Victims of war, because sometimes all you have is fragments of people, unfortunately. And as a result, you can do DNA testing to determine family relationships with these people. Large-scale disasters, DNA evidence is really important. Biodiversity in terms of biology, ecology, and genetically modified crop usage. And then sometimes immigration disputes where families are being separated, and a lot of times that is hard to kind of track. But if you use DNA, you could get the kids back with their biological parents. Um, this was not initially intended for these purposes. They were intended for treatment of disease by the medical field, but quickly learned that they were powerful in other instances as well, and so now they're used in all these different cases. Small amounts is all you need. You have trace evidence in the form of you know saliva, blood, semen, skin. Sometimes hair follicles from the root would have enough DNA that you could use them. Body cells from tissue, and sometimes urine. Not always. So there's quite a lot of different sources you can use to get a DNA sample. Obviously, blood is probably one of the best because while red blood cells don't have DNA, white blood cells have plenty of it. Um, but it's been lifted from all sorts of different sources. So there's technology out there that allows the scientists to look at even bite marks and lift DNA from inside the bite that they can use to match the criminal based on the DNA in the saliva that was found in the bite mark. We'll get into the structure and function of DNA just a little bit. I don't want to go too far with this because this is kind of outside the scope of this class. DNA is one of the 4 biological molecules found in the form of chromosomes. Typically, though, it's not a chromosome within a cell. Typically, DNA is kind of a loose granular form called chromatin. Chromosomes only form up when the cells themselves are going into cell division. They're found within the nucleus. So only cells that have a nucleus like this in our bodies will have DNA. In bacteria, the DNA is kind of free floating, so it's a little different. Normal humans have 23 pairs of chromosomes. Egg cells and sperm cells have half of that because they need to be able to combine together to form a full 46 total. Each chromosome has regions on it that are broken down. They're called genes. Each gene controls a trait or sometimes more than one trait if they're working in conjunction with other genes in the body. They vary between individuals. This is what makes us all different from each other. Genetically, DNA, or structurally, DNA has a significant structural basis to it. Every single nucleotide contains one of 4 different nitrogenous bases: adenine, thymine, guanine, and cytosine. Also will contain a nitrogen, or sorry, not a nitrogen base, a sugar and a phosphate that are part of the backbone, which in this picture here would be the green strip that goes down the outsides. They always form using rules of base pairing. Otherwise, you get bubbles in the DNA, which is bad. Adenine binds only with thymine. Cytosine binds with guanine. This is what we call complementary base pairing. So if you look at the bottom, the sequence ATCTGC, the complementary strand to that is going to be TAGACG. Okay. Because again, if you have an A on one strand, you have to have a T across from it on the other strand. Now, when DNA forms up, it forms naturally into the shape of this double helix, and that double helix is actually caused by hydrogen bonds that go between the adenine and the thymine and the cytosine and the guanine. And if you look closely, you can see that adenine and thymine form 2 hydrogen bonds between them, where cytosine and guanine form 3. The sides, the backbone, again, I already said, phosphate and sugar, and they just repeat. The rungs that go across the middle are where the nitrogen bases are found. They help us. All of this, it's the sequence of this that determines the form and function of the protein that's being produced by the gene that is being made up by this DNA. So here's a kind of an expanded view. You can see the sugar and the phosphate in the backbone, and they just repeat over and over and over again. Then you have your nitrogen bases across the middle and the twist of the DNA. So this is a really commonly well-studied feature. There are different forms of DNA, but not in humans. DNA that is found in the chromosomes is our nuclear DNA. Um, these are only found in the autosomal cells, the cells of their body that are not used for reproduction. Um, you get your nuclear DNA from both mother and father. You get about half from each. Mitochondrial DNA is a little different. It's only found in the mitochondria, and it's a circular form of DNA, very similar to bacterial DNA, but you only inherit that from your mother. So this is how um, matrilineal studies can look at how how people have inherited their mitochondrial DNA through generation after generation, because it doesn't mutate as fast as nuclear DNA typically. Um, you can get your DNA from both of these. Personally, I've worked with both forms of DNA. Nuclear DNA is easier to extract, but mitochondrial DNA, again, gives you a better picture of long-term evolution because it's slower to mutate. Um, when sperm and egg come together, obviously the two half sets of DNA merge together to form one whole cell, which then starts to divide, and that becomes human. The human genome itself, that is the sum total of all the DNA. We have about 3 billion base pairs in our genome. Not all of that works to encode a protein. There are introns and exons in our genetic code that don't, the introns don't code for a protein. The exons do. Introns are removed and the exons are joined together. It's kind of a complicated process. And again, we don't really need to get into that. But the purpose of the whole entire code is telling the body how to make the proteins that are needed to do the work that goes on in the body. Again, exons, they're only about 1.5 to 2% of the entire genome, and there's only about 24 to 25,000 different genes in our genome. Despite the fact that we have 3.3 billion base pairs of DNA, we're only using a very small section of that. The rest of it is introns that get removed before the proteins are created. Used to be, people thought that introns were junk DNA, but they're really not. They're really important in things like gene splicing, but also they give us a source of variation when adaptations need to be made in the process of evolution. They're really, really important in forensic science because they don't tend to mutate as fast because they're not being used. So they tend to stay the same. And they have regions in them that are really, really handy for forensic science. We'll get into that in a minute. Most human genome, it's the same. If you look from one person to the next, you're probably going to see, 99% of their DNA is the same because there are things that every human needs in order to survive. So you're not going to see a lot of variation in the coding regions. The exons are going to be very similar from person to person. There will be some differences, but not as much as you're going to see in the intron regions because they're not as important for day-to-day life, so they can change without having serious impacts on humans' ability to do things like create, proteins that are necessary for breakdown of fats and things like that. A lot of times the introns will have segments that are repeated, and they can be repeated many, many times, like 15, 20, lots of times. And if you see sequences like that, they can form unique patterns in different individuals. So that's what we call a polymorphism. They're the non-coded DNA regions that contain patterns that are unique to individuals, and they can be used to identify a person forensically. So here you see a couple of different types of things. We have two different types that we have used. The current standard in forensic science is the use of STRs, which are short tandem repeats, but VNTRs, variable number of tandem repeats, have also been used. If you look at the image here, what you'll see is that we have these short tandem repeat sections, CTA, CTA, CTA. It's being repeated here. In man 1, it's repeated a certain number of times. There's 5. But in man 2, there's 6. And in man 3, there's 7. We use multiple STRs to identify people. I think at this time we use 13. And the odds of having the same exact STR pattern from person to person are incredibly low. So it's really, really powerful for identification purposes of individuals, based on their STR patterns in their DNA. They're both used for our fingerprinting process, and they can be used for things like tissue matching. The thing is that the STRs, the VNTRs, the information that you get from them is incredibly sensitive. So you have to use really careful process to get this evidence out and get it to the court. But you can look and see that the patterns from crime scene here and suspect number 2 match up down through the whole sequence. And so we would know based on this that suspect 2 was definitely at that crime scene, and that would be, well, I shouldn't say definitely. Suspect 2 was most likely at that crime scene, and that is very strong evidence against suspect 2 saying he wasn't there. We can also use it for inheritance matching. So we want to look at familial relationships. Obviously, again, you can look at the mother's DNA, the child's DNA, and the father's DNA and try to compare them. And if you look at this first one here, you can see mother, she's here, the child is here. They share a band right there, mother and child, so they're related. But also father and child share a band right here. So this is definitely evidence that this is the father of that child. Same thing over here, except, it's the same thing. In this one, there is a match here also. So again, all 4 of these are showing that the father here, the alleged father, is likely the father of that child. So if you look at this pattern, these are the children: daughter 1, daughter 2, son 1, son 2. Mom is definitely related to daughter 1 because they share bands, and dad is also definitely related to daughter 1. If we look at daughter 2, these red bands here do not match up to the father. So this is not his daughter, but this is the daughter of the mom in question. And son 1, again, bands match up. Okay, so son 1 is the father's child, but son 2, the bands do not match up. So we can look at this and see that in daughter 1, mom and dad are the biological parents. In daughter 2, mom is a biological parent, but has a different father. In S1, same as D1, both are the biological parents. And then in S2, we're not seeing any patterns from mom either. So this is not her son. So often the amount of sample for DNA can be very, very small. So we consider it a form of trace evidence. It's hard to work with because when you collect it and you use it, you're basically using it up and it's gone afterwards. If we need to get more evidence, we can take and use a process called polymerase chain reaction, which will take a tiny sample of DNA. I mean, we're talking like a pinprick of DNA here, and you can make millions, not thousands, millions of copies of the DNA that's present there in a very short period of time and allow enough of the sample there to be analyzed. The process works like this. You have to have some equipment for this, and we did have this in my classroom for a while this year. Hopefully we'll have it again next year. We'll see. This is what we call a thermal cycler, this machine here. And what it does is it runs through heat changes. And each stage of heat change has a different function in terms of what it does to the DNA. The first one is what we call denaturation. You heat the DNA up to pretty close to boiling point. What that does is it drives the DNA strands apart from one another and exposes the base pairs. Then we cool it down to a lower temperature. Um, Depending on what we're doing here, it depends on the primers that we're using. 48 to 72 degrees Celsius. What that does is it allows the primers, which are specialized sequences of DNA that match up to the ends of our DNA sequence that we are trying to magnify. They will bind to that and give the DNA kind of like a building block to start from. And then we start with that's called annealing. And then we go through the extension phase. Extension, you heat it back up a little bit. DNA polymerase from a bacteria that was discovered in the hot springs that can work at a higher temperature than human DNA polymerase, which would only work at about 37 Celsius, will then drive along the DNA in both directions and build new DNA strands matching the sequence on the blue strand that you see here. So because these are complementary base pairs, what you end up with are exact copies of the DNA every single time. This double helix here will be identical to this double helix here. You can, over the course of 2 to 2.5 hours, you can have several billion copies of your original DNA sequence, the sequence of interest that you have primers for. And that's where the STRs come into play. We know what the STRs are, and we know what the DNA on either side of the STRs looks like. So we can make primers for either side of each STR, and then we can magnify the STRs greatly. And that will then give us a DNA fingerprint that can be used. It's a very interesting process. DNA is really sensitive. It's hard sometimes to collect it effectively. And this is if DNA evidence is shot down in court, usually it's because it wasn't handled properly. Because a little bit of contamination or cross contamination can cause a lot of problems. So a lot of times when they're running these DNA sequences, they will run crime scene, suspect, and evidence collection person in the same sequence to be able to rule out this DNA over here came from the evidence collector. This DNA over here came from the crime scene. This DNA here came from the suspect. So there are some kind of rules that are in place for this. Oftentimes, people who are collecting DNA will wear a mask or a face shield so that if they do sneeze or cough, they're not going to contaminate. They have to wear disposable gloves. They have to sample with instruments that can be disposable. Air dry the evidence before packaging. You can freeze DNA for quite a long time, actually, and be able to use it. And then you have to make sure you keep it out of the sunlight because we all know UV rays damage DNA. You can then collect DNA from your suspect or from the researcher, the evidence collector, through a couple of different ways. You can take blood draw. You can do a swab from their cheek or something like that. And then they're going to take that and they're going to cut it up. Well, if they don't have to, but they will often cut it up into fragments because the fragments are easier to work with. And then they will amplify through PCR to look at the patterns and be able to generate a DNA fingerprint. Gel electrophoresis, basically what you do is you take your samples and you load them onto a gel. You put that gel into an electrophoresis tank, which is what this is right here, the kind of darker gray. You have a negative electrode at one end and a positive electrode at the other. You float it. You put the gel in a buffer, which is going to transmit electricity, and you turn on a power source. And what it does, because DNA is negatively charged, it drives the DNA from the negative electrode because like charges repel. It drives the DNA from that negative electrode toward the positive. Size is important here because the smaller fragments are going to move faster than the larger fragments. So this is a pretty important process. Then you get a gel that looks kind of like this up here, where you have fragments at different points in the gel. And you can then use that as your evidence. Basically, take a picture of it, and then you can share it. So this actually is what a typical gel looks like. It's been stained with a stain called ethidium bromide. When you hit it with UV, it glows really brightly, and it's really sensitive. You can see really small fragments on here based on the fact that they have the ethidium bromide in them, because the ethidium bromide, what it does, it gets right into the DNA and bonds to the DNA. And then when you hit it with the UV light, it fluoresces. So it's really an interesting process. And you can see here this lane called marker DNA. These are sizes that are known. So this could be like 100, 200, 300, 400, 500, 600 base pairs. Using this, we can then say, this is 300 base pairs in size. And if you were looking at the crime scene, you'd be looking for matches in between the different lanes of the gel. So again, we're looking at different loci here, locus A, locus B, with different tandem repeat regions. You can see here, we're looking at GC repeat. How many of them do they have? And then we're going to load these onto a gel down here. And what you see is after electrophoresis, the banding patterns between the two individuals here are very different. They don't share any commonalities between them. So this is actually a really, really useful and powerful process that allows us to determine very effectively, did these samples come from the same two people, or are they from the same, are they from different people at the crime scene? You can get really complicated with these, but typically when we're doing this at the high school level and looking at these, we tend to use simplified versions of these STRs because they can get really complicated. But you have to make sure you're loading in, the victim's DNA, because the victim was clearly at the crime scene, any of the suspect's DNA, and then you're also going to put in oftentimes the scientists who collected the DNA, put theirs in there too, and then run it with a positive control to make sure, because the positive control needs to be there to allow you to say, we did this. You also should run a negative control. We did this and it worked. That way, that what you're looking at on this gel is actually scientifically supported. So then we look at the bands and hope that we find matches in the DNA. If we don't, then this is not really forensically supporting any kind of suspect against, in our case. So in the past, we've talked about the Innocence Project. DNA is being used very, very often by the Innocence Project now to do things like free falsely imprisoned individuals. And it is this technology that is actually being used a lot of the time now to help prove that people who are wrongly imprisoned should be let go. And it's actually really interesting. For those of you who aren't in my class that just happen to be watching this video, I strongly recommend that you go look at the Innocence Project website. It's innocenceproject.org, and take a look. It's really interesting how many people there are that are in jail and have been in jail for a very long time, who have been in jail based on very weak evidence and probably should be released, because there's not strong enough evidence to really keep them in prison as long as they have been. The other things that we can use this for, again, determine maternity, paternity, identify human remains, eliminate suspects, or to provide a match to a suspect at a crime scene. There are DNA databases. CODIS is the United States federal version. It's the Combined DNA Indexing System. There are millions of profiles of DNA from felons of violent crimes, and sometimes CODIS can actually connect a person from one crime scene to a whole string of other ones. And in fact, people in prison have actually been connected to previous crimes by matches based on what's in CODIS. So it's a very powerful thing. We also have DNA profiles of everyone that's in military service, in case they are killed overseas or something like this, or victims of war. Um, then we can identify the remains using DNA. DNA profiles are always taken from rapists, murderers, child abusers. Other types of violent crimes are also included in this. And that would be about it for today. I hope you enjoyed this. And if anybody has any questions, hit me up on my email. We'll see you all later.